Welcome to our webinar, Setting Up 3D Spheroid Assays Using High-Content Imaging.
I'd like to introduce you to the speakers and provide you with a high-level agenda. My name is Jane Hesley. I'm an application scientist in cellular imaging, and I'll start off with an overview of 3D imaging, including acquisition and processing of spheroid images, and I'll touch on the analysis of the images. Then Oksana Sarenko, one of our research scientists, will explain an optimized method for performing a high-throughput compound screen on cancer spheroids. You may submit questions as we go along, and then at the end, Grisha Chandy, the cellular imaging product manager, will moderate the questions.
During the webinar, to ask a question, press on the Q&A button on the toolbar to expand the Q&A window. Type in your question, choose All Panelists, and click Send. If you can see the webinar but are having difficulties, dial the technical support number below or type a question and send it to all the panelists. The assays in this presentation were developed using the MetaXpress Micro high-content screening system in conjunction with our latest MetaXpress software release. For those who are not familiar with the system, MetaXpress Micro is equipped with features such as laser autofocus for reliably capturing an in-focus image every time. It has the capability of housing 4 objectives and 5 fluorescence filter cubes at once, and can acquire images from slides to single-well plates to 1536-well microplates. Some features that are essential for spheroid work are its capability to be integrated with robotic plate handling, to acquire time-lapse images in time spans of seconds or days, which is where the importance of full environmental control comes in. You can also acquire transmitted light images for non-stained samples and set up to acquire multiple images in the z-axis to interrogate cells throughout your 3D sample. Why perform 3D imaging? This introduction is pertinent to people starting to explore spheroid imaging in a higher throughput manner, such as researchers in the fields of oncology or in stem cells. 3D co-cultures of cells, such as liver or kidney with their supporting cell types, often called organ in a dish, are being used to test both on-target and off-target effects of drugs as a model that more closely represents a whole organ. This image of Hebergen's hepatopac plate shows one island of hepatocytes arrayed in a field of supporting fibroblasts. Also, various cancer cells grown into a 3D structure are a popular model for tumors, such as this image of a spheroid taken at 10x magnification. Spheroids have a role as samples in drug discovery, even at the screening level, and in testing specific cell or organ toxicity, and in personalized medicine, where cells from a biopsy can be used to grow tumor spheroids, and then anti-cancer drugs can be tested to find the right drug treatment for that patient. One reason scientists are turning towards the use of spheroids is that they may react to compound treatments differently than a flat 2D cell culture. There are numerous reports showing that the effective concentrations of drugs may be higher or even lower when tested in a 3D system versus a 2D system. This image illustrates the difference in a dose response in a 2D cell culture system on the top versus the same doses used to treat the spheroid arrays in the bottom images. This study, using human prostate cancer cells, showed that a higher dose response of the anti-tumor drug gemcitabine was required to slow proliferation of cells if the cells were growing as spheroids. I'm going to discuss a few of the ways to grow cells into a spheroid. The first is to give the cells a scaffold to grow on. Microplates and slide surfaces have been manufactured to contain 3D structures or fibers that cells attach to and which encourage cells to adhere to each other. Alternatively, the surfaces are treated so the cells cluster into arrays or islands in specific areas of the plate bottom. This image shows an array of stained spheroids on a sellable oncology plate from Toyo Gosei. Another way to provide cells with a 3D area to grow is by combining them with an extracellular matrix, such as the commercially available gel matrices Agarose, Matrigel, and Sigel. This is an image of PC12 cells growing throughout a thick layer of Matrigel in a 96-well plate. A third method for creating a spheroid is by simple gravity. For example, the popular hanging drop method as seen in this illustration by 3D Biomatrix. A number of cells is pipetted into a small hole in the specially engineered plate. Surface tension keeps the drop hanging beneath the plate, poised above a carrier filled with liquid to prevent evaporation. The cells sink to the bottom of the drop and within a day have formed a spheroid. Small amounts of solution may be removed or added to the top of the drop through the hole so the spheroid may be treated and stained. This process can be automated and the spheroid can be imaged using MetaXpress Micro either in the hanging drop plate or after forcing the spheroid to drop into a separate imaging plate. The results Oksana will show in this webinar were achieved with this last method where cells are cultured in low adherence round bottom wells. In this montage of transmitted light images, you see excellent reproducibility in the 2-day-old spheroids growing in a 96-well round bottom plate. The high-content workflow of using spheroids is similar to that used with any cell-based assay, although the times required may be longer. In our examples, the cells were seeded and allowed to grow undisturbed for at least 2 days. Then, either a partial medium change or addition of the compound was performed, and spheroids were incubated for an additional 1 to 3 days. Live spheroids were stained and imaged either with or without a careful wash, depending on the detection reagent used. MetaXpress Micro was used to image the wells, and the resulting images were analyzed using MetaXpress software. Often when interrogating a 3D spheroid, multiple images of the object are taken in different vertical z-planes. Here I show a screenshot of the Z acquisition window in MetaXpress software. You set up the number of planes and step size for each wavelength, and the total range or height of your stack is calculated here. The ruler and the spaces for entering the number of steps are interactive. You can change the acquisition parameters by typing in a number or by dragging the top or bottom points on the ruler, and the numbers will be updated. To make it easy, these adjustments can be made while you're looking at the spheroid in live acquisition mode. You may notice that in the different planes some cells are in focus, while other areas are out of focus. After collecting a stack, you may save each plane or else collapse all the planes into a single image for ease of analysis and to reduce the amount of computer storage space used. There are several mathematical algorithms that can be used to generate the 2D projection. Here is the 2D best focus projection generated from the stack you saw on the previous slide. Best focus identifies the highest contrast pixels in the stack to keep for assembling the 2D projection. Another option is maximum projection, where the pixels with the brightest intensity in the stack are kept in the 2D image, and all others in the same XY space are ignored. The 2D projection you choose between best focus, maximum projection, minimum projection, and sum is generated at the time of acquisition, but if the z-planes are saved, they can also be generated from the stacks later, for example if a different 2D projection gives a better image for analysis. If you use a 2D projection to generate an image with the most in-focus area, more of the cells in the spheroid can be analyzed. At the bottom of the z-stack, the nuclei in the center are well in focus and easily segmented by the software, as seen by the yellow objects on the teal spheroid mask. Towards the middle of the spheroid, the nuclei at the edges come into focus. By collapsing the stack, both areas are combined, and analysis can be performed on a larger population of the cells. Our software allows this analysis to begin as soon as the images are generated, and also, for even faster results, can be sent for parallel processing of multiple sites or wells at one time with PowerCore software. Spheroids can be analyzed using our pre-configured software modules such as LiveDead, Cell Health, Multi-Wavelength Cell Scoring, or many others, or you may opt to set up your own analysis using a custom module with all of the typical image and object processing features available in a user-friendly drag-and-drop format. In Custom Module Editor, you can design and name your segmentation masks and specify only the information you want to be output. Any of these analyses methods can be saved and shared. This shows a heat map in MetaXpress of an analyzed plate with the number of cells positive for apoptosis overlaid. In summary, the MetaXpress software ties it all together since it's used to acquire your images either as a simple single plane or multiple planes, or as a single endpoint or multiple time points for a few minutes or up to a few days if your instrument has the environmental control option. Then MetaXpress is used to analyze the images, and all images and data are stored in our provided database to be recalled from any networked computer. Here's a pictorial depiction of what I just said. ImageXpress acquires images and stores them in a database where they are analyzed by MetaXpress, and the data is stored with the plate where it can be accessed by our data mining software called AcuityXpress for generating curves and identifying active compounds. In addition, third-party imaging systems images can be imported into the database for analysis in MetaXpress and then data mining using AcuityXpress. Next, I will pass control to Oksana Serenko, who will share her development of some high-content screening assays in cancer spheroids. While we switch presenters, I'll remind you how to submit a question. Pull down the Q&A window, type in your question, choose All Panelists, and then click Send.
Thank you, Jane, for your kind introduction. So, hello everyone who listens in this presentation. I am Oksana Serenko, research scientist at Molecular Devices, and my today's presentation is about development and optimization of spheroid assay that can be used for compound screening. Cell models for assays becoming more and more complex in order to better mimic in vivo environment and provide greater predictivity for compound efficacy and toxicity. There is an increasing interest in using three-dimensional assays for modeling cancer and tissue biology. Three-dimensional in vitro models believed may span a gap between two-dimensional cell cultures and whole animal system, and may provide better perspectives on behavior, cells, developing tissues and organs, and growing tumors. The goal of this study was to optimize cell culture and develop new imaging and analysis methods that can be used for compound screening. High-content imaging is a very important tool for using in complex cell model, including 3D models. In this picture you can see spheroids, either controlled or treated with different compounds, and you can see dramatic changes in cell morphology, which may provide very interesting information about the effect of different compounds. 3D cultures provide maybe more accurate prediction of the activity of anti-cancer drugs, but there are also a number of challenges, which is regard of cell culture, staining, image acquisition, and image analysis. Various techniques were used to form spheroids. The most common is hanging drop techniques. But there are several challenges for cell culture, media change, and compound addition, usually those manipulations done using liquid handlers. We have used ultra-low attachment plates that allow much easier cell plating, media change, and compound addition. The workflow is similar to the processing of standard cell culture. You can see the movie of spheroid formation over weekend. So plated cells form the clump first, and then it becomes more dense and forms spheroid. We have done optimization of spheroid culture for three commonly used cell lines. We show here dependence of spheroid width from the number of plated cells. Two large spheroids are more difficult for imaging and for dyes and compound penetration. Two small spheroids may have irregular shape or inconsistent size. Optimization can be done in real time using transmitted light imaging. Image analysis allowed to find spheroid object and take some measurements of spheroid size, like diameter and area. You can also monitor spheroid size over the time and also effects of compound treatments. We show here dependence of spheroid width from the number of plated cells for 3 different cell lines. On this slide you can see an example of 96 well plate with spheroid cultured for 7 days. Note that the size and shape are very consistent and standard deviation for spheroid width is less than 10%. The assay is convenient and feasible for the high throughput screening. So plating and media addition similar to regular cell culture, and spheroids are cell-centered, which is very important for imaging. You can capture spheroids in single image, 10x or 20x objective. (10x or 20x magnification allow enough resolution to look at different cell markers. There are other challenges for spheroid culture protocol, like dye penetration may be difficult, especially if you use antibodies. In our assay, we used Hurst for nuclear stain, calcine AM for viability, and also ethidium homodimer for dead cells, which stains nuclei of cells with damaged external membrane. So our staining protocol includes one-step addition of 3-dye mix. This is live cell staining and no washes required. It is important to keep in mind that timing for staining spheroids and concentration of dyes usually higher than in traditional assays. It takes some time for dyes to get into spheroid, and also it is better to use higher concentration of dyes. We allow about 3 hours for staining reagents to penetrate into spheroids. Usually it's done in the incubator. It is better to optimize your protocol to reach desirable intensity of dyes, and here we present optimization graphs for our assay. Imaging of 3D cultures and spheroids present other challenges. First, we have to use this stack of images, one side per well. It is essential to capture large objects without splitting, and it is very convenient when spheroids are in the center. Second is how to compare different spheroids. On the top panel, there are several spheroids presented that have been treated with different compounds for several days. Taking single image may not work if spheroids become different sizes as a result of treatment. You can see it on below diagram. One way is to analyze all images, but it is very helpful to use 2D projection from several slices, best focus. 2D projection allows to compress information from different slices and allows easier comparison between spheroids. For image setup, we use 10x or 20x objective, one side per well. We use this stack, 7 to 9 images, could be more if desired, and best focus 2D projection. At present slide, you can see the nuclear count across several Z slices plotted against distance from the plate bottom. So the projection image allows comparison of spheroids with different phenotypes and also reduces storage and time for analysis. The movies present this stack of individual images and best focus image. Of course, the image on the right is more easy to analyze. This is a screenshot of spheroid plate treated with 6 compounds with increasing concentration. Live cells stained with 3 dyes. And here you can see best focus projection of different spheroids. So you can observe difference between controlled spheroid and also spheroids treated with Staurosporine or doxorubicin. This assay is a perfect example of phenotypic screening. You can see here spheroids treated with different concentration of several compounds. There are very dramatic changes in spheroid phenotypes. For example, you can see decrease in spheroid size or disintegration of spheroid, or you can see decrease of live cells and increase of dead cells, which are stained in red. The assay allows not just to monitor decrease in size, but also characterize specific changes in spheroid phenotypes. There are several challenges for analysis: how to define parameters which best characterize different phenotypes, and how to quantify diverse responses with multi-parametric analysis. For the analysis, we apply nuclear count in spheroids, as shown on the top on this mask, also cell count and cell characterization based on expression of different markers. Here there is cell scoring for live cells, in this case calcine AM positive cells, and here is cell scoring for dead cells, Etidium homodimer positive. Another example of spheroid assay is apoptosis. Spheroids treated with compounds that cause apoptosis and then stained with Chi-space reagent and also Hurst nuclear dye. You can clearly see increase of green cells in spheroid treated with Staurosporine, and you can quantify the numbers of intact cells and also apoptotic cells by using cell scoring analysis. We use custom model editor to identify spheroids, usually in DAPI channel, and then we apply nuclear count and cell scoring algorithms. You can see several examples here that show the mask of the objects and also masks of individual cells. For analysis, we define number of readouts that characterize morphological changes, like area or diameter or spheroid width, and also we characterize total cell count in spheroids by nuclei count, the number of live cells as Etidium homodimer negative, a number of dead cells, Etidium homodimer positive, as well as number of calcine AM positive cells that defines metabolically active cells. This bar graph shows measurement for 3 examples of spheroids. So you can see decrease of live cells, increase of dead cells, decrease of total cells or calcine AM positive cells, as well as decrease of spheroid width. Those different readouts allows to characterize specific responses to compound. The number of live cells or other readouts can be used to define IC50 for different compounds and compare compound potency. So here presented IC50s in nanomolars for number of tested compounds. Finally, I would like to summarize that this assay can be generated with very good reproducibility. We define protocols for multiple staining, and also we show that spheroid phenotypes can be described using image analysis with custom model editor and multiple measurements. Those different measurements provide expected dose responses to known compounds and can be used to compare compound efficacy. Thank you for your attention. Please follow up with your questions. And I would like to acknowledge Trisha Mitlow and Evan Cromwell for great help with this project. Thank you.